In this video, we will be outlining our findings in regards to Crouzon syndrome, the disorder caused by mutations in the FGFR2 gene, and how we use bioinformatic tools to develop our understanding of its biological mechanisms and treatment. Crouzon syndrome is a genetic disorder that occurs from a missense mutation in the FGFR2 gene. The syndrome leads to premature skull suture fusion, an altered face shape and features, and eye abnormalities, all which have a drastic impact on life quality. The FGFR2 gene normally codes for the FGFR2 protein, which is necessary for osteoblast proliferation and differentiation, which are vital steps of bone formation. The mutation leads to overactive signaling from FGFR2, causing the downstream pathways RAS/MAPK, PI3K/AKT, and PLC to be excessively activated. These pathways are responsible for proliferation, differentiation, and cell survival, which therefore leads to abnormal bone development. Bioinformatics tools can be used to understand the mutation, where it occurs in the gene, and how it affects each biological level, using this to develop potential treatment options. We will explore this by looking at ORF and XPARSI as the initial insight into the gene. Then we will look at ClinVar, which explores known mutations that are linked with Crouzon syndrome. Following this, ProCyte analysis will be discussed to understand cellular mechanisms of the disease. And with all this insight, we will discuss how bioinformatic tools can be used to develop personal treatments, including already developed and possible treatments.
Now we'll look at XPAXI here. So basically what happened in XPAXI was we had a DNA sequence which was run through XPAXI and it generated 6 open reading frames, as you can see here. So from this analysis, we can see that 5'3" frame 1 was most likely to match that of FGR2 due to having the longest amino acid sequence. And also the red regions represent the stop regions within the open reading frames.
In our bioinformatic breakthrough booklet, we use the ClinVar database to gain an understanding of the previously categorized pathogenic variants in the FGFR2 gene that had been linked to Crouzon syndrome and other craniosynostosis disorders. Through using ClinVar analysis, it was evident that the most common mutation type that led to Crouzon syndrome was missense mutations. None of the mutations were frame shift or nonsense, and instead led to a single amino acid change. ClinVar was beneficial for studying these mutations as it allowed for observation and analysis of known variations in people who have Crouzon syndrome. These mutations can be compared to each other in style of mutation and effect on phenotype, particularly to identify if the mutations are pathogenic.
Referring to published literature, we found that many of these pathogenic missense variants occur in or near the functional domains of FGFR2 protein, most commonly Ig-like domain 3. With this in mind, we conducted protein domain analysis using ProCyte to gain a deeper understanding of the functional domains in FGFR2 and how mutations can affect their structure and function. By inputting the amino acid sequence, ProCyte is able to output the series of functional domains in protein through comparison of conserved sequences of known domains. In FGFR2, 3 Ig-like domains and a protein kinase, or more specifically, a tyrosine kinase domain, were identified. As mentioned earlier, many pathogenic variants stem from disruptions to the Ig-like domains. These domains are located extracellularly and serve to form the ligand binding site of the receptor. In FGFR2, the ligand binding pocket is located between the second and third Ig-like domains. Missense mutations that disrupt the amino acid sequence forming these domains can cause changes to the structural arrangement, and as protein function is closely linked to its structure, this often has a flow-on effect where the overall protein function is also modified. In Crouzon syndrome, the FGFR2 protein and the downstream signaling pathway are overly active, causing the premature function of cranial sutures. These types of variants are known as gain-of-function mutations. This overactivation suggests that the missense variant causes a change to the structure and the function of the ligand binding region, such that fibroblast growth factor binds with a much higher affinity, or alternatively, there's a change to the protein making it constitutively active, not requiring ligand interactions at all to form the functional homodimer. Both of these result in the same molecular outcomes of overactivated downstream signaling of cell growth pathways, resulting in increased osteoblast proliferation and differentiation, causing cranial suture fusion and abnormal skull structure that's observed in those affected by Crouzon.
To assess downstream pathways and measure their activity, proteomics was a beneficial tool that was used. Mass spectrometry detected altered expression of signaling proteins and changed protein interactions associated with overexpressed FGFR2 signaling. This provided insight into how mutations within the FGFR2 gene, particularly those leading to altered activation, affect downstream effectors. Literature was used to further understanding of how mass spectrometry can be used to analyze, particularly looking at the study iTrack-based proteomics analysis reveals possible target-related proteins and signal networks in human osteoblasts overexpressing FGFR2. By using iTrack-based quantitative proteomics, isobaric tags can be used to label and measure proteins simultaneously using mass spectrometry. The study identified 149 differentially expressed proteins that occurred during signal transduction, highlighting how proteomics is a powerful tool for identifying downstream proteins and adapting this for targeted drug development. Using these tools allowed for a deeper understanding of the cellular mechanisms behind this disorder, helping to build a more comprehensive overlook on Crouzon syndrome and its pathology. And this can lead to innovative breakthroughs in treatment options for those affected.
An example of a treatment approach that targets the overexpression of downstream FGFR2 pathways is a personalized medicine approach that, while not yet in clinical trials, has shown potential in reducing the effects of pathogenic variants. This is the allele-specific knockdown of FGFR2 protein using small interfering RNA molecules. The basis of this treatment is to halt the production of mutant FGFR2 protein by introducing complementary siRNA molecules that are specific to the patient's mutation. These siRNA molecules can bind to the mutant mRNA with full complementarity, such that the mRNA is redirected to be degraded by the cell instead of being translated. This method was found to successfully downregulate FGFR2 activity in affected cells, as well as showing promise regarding the reprogramming of mutation-positive osteoprogenitor cells.
From our booklet, we found that for Crouzon syndrome, a promising treatment was the CRISPR-Cas9 editing, which works by using a guide RNA, which recognizes a target sequence and uses a double-stranded break to edit and remove the gene. This potential treatment is possible as there have been specific mutations logged on ClinVar that have led to Crouzon syndrome. This would mean that guide RNA could be produced specifically for these variations that are known. This would not address all mutations that have led to Crouzon syndrome, only those logged on ClinVar, but is a strong foundation for the CRISPR-Cas9 technology.
Working in a team is always challenging, as different people will bring different perspectives and working styles to the table. As a team, we decided to set out plans and establish clear communication lines to combat possible conflicts. We created a group chat on Instagram where we could communicate with each other regarding meetings and workload, and we also used a shared Google Drive where we could collaboratively complete work. Within this Google Drive, we had copies of all relevant information for Map 2, including instructions and assistance documents that could all be easily accessed and referenced. Our report was compiled on a shared document, making it easy to track each other's progress throughout the project. In this document, we were also able to give each other feedback using the comment or suggest functions to ensure that the final breakthrough booklet was cohesive between sections. This route for feedback was also supported by regular online meetings where we could discuss progress and give more detailed feedback. In these meetings, we'd go through the entirety of the document, figuring out what tasks needed to be completed and allocating team members where relevant. This ensured the entire group was on the same page in an attempt to prevent future conflicts. In these meetings, we also set future timelines and internal due dates, helping to manage group expectations, and of course, where necessary and when the group agreed, we could shift these timelines in order to ensure high-quality work. In the meeting planning for this video, we discussed what we thought were the most important insights from our research and how they could link to each other to create narrative connections. We then split up the sections we decided on so individual members could write scripts and create PowerPoint slides. The factors of consistent communication, regular meetings, and shared documents were the base that we built our teamwork approach on in order to effectively produce our bioinformatics breakthrough booklet and this video.
In conclusion, our bioinformatic investigation into Crouzon syndrome has allowed us to connect molecular genetics with real-world clinical outcomes. By analyzing the FGFR2 gene, we discovered how specific mutations can alter the protein structure and disrupt normal signaling pathways involved in cranial development. Through protein structure modeling and protein domain analysis, we gain insight into how these mutations influence the receptor's conformation and interactions within key signaling networks. The transcriptomic and proteomic data then reinforce these findings, showing how changes at the molecular level translate into altered expression patterns and cellular behavior. In the proteomics section, ultimately measured the regulation of the FGFR2 protein, which contributed to the excessive growth of FGFR2, thus contributing to early suture development. Bringing these sections together, our research highlights how each layer of bioinformatics from genomics to proteomics contributes to a more complete understanding of disease mechanisms. These insights not only deepen our understanding and knowledge, but also open the door to emerging precision therapies such as allele-specific knockdown and CRISPR-Cas9, which represent promising future treatments for patients with Cushing's disease. Overall, this project has demonstrated the power of collaboration and integration analysis in uncovering complex biological relationships. Our team's collective effort turned extensive data into a clear, structured, and meaningful story, one that bridges fundamental science with potential therapeutic innovation. Thank you.